Right, it's recording now. Let's try and share screen. And now this is the exam session, study session for plant cell bioprocessing. It's a university course that I take in India. It was online. It's coming up soon. It was a shortish course. It was only an 8-week, not a 12-weeker. So it was good. But and what I do, I've got on, can you see me there on the screen? I've got a little box thing that I put in front of it, because this one has the answers are pretty much on the left-hand side. So I put a little box down on the bottom of the screen like that, just so I can't see the answers, and it covers up the little box thing with selections. So that'll help a hell of a lot too. So if you just get a little tiny box and put it down on the bottom of the screen and just fit it there, and right there, you can't see it, but that's where it is. So it'll help. And that's what I've done. So I can try and not, so which of the following organelles is responsible for storage of fats and lipids? It's the eleoplast. Which of the following plant cells typically provide mechanical support and form part of the secondary cell wall? That's the sclerenchyma. Epidermis forms part of the tissue systems. It's the dermal tissue. It's on the edge. Vascular cambium and cork cambium are examples of which of the following meristems? Oh, it can't be the top or the inter. It actually can be a little bit there, but it's the lateral. Theory of water transport in plants is the cohesion tension theory. Which of the following is an example of a C3 plant? Sunflower. The light-dependent reactions in photosynthesis fall under the Kelvin-Benson cycle. False. In C3 plants, photosynthesis, carbon fixation, and Kelvin cycle occur in single. They do. Spin out, eh? Is that it? It is true. Plants overcome photorespiration by utilising rubisco as its carbon dioxide fixing enzyme. False. We'll use something else as well. Which membrane barriers are to be surpassed generally in cultured plant cells to be released in secondary metabolites? Tono and plasma membrane. Yeah. Sweet. Week 2. 70% ethanol is generally used as a something during sterilisation of explants. It's a wetting agent. Transfer of callus to a fresh medium should be done before the something phase, or you want the good growth rates. So the log phase is where there's heaps of exponential growth, so it's got to be before in that, so it's stationary phase. Which of the following methods is not generally used for determining growth in plant cell cultures? Optical density at 600 nanometers, you don't use very often. Callus induced in vitro explants is generally composed of parenchyma cells. They're the ones that have nothing, no direction yet. A non-sexual development process by which the bipolar embryo for somatic tissue is termed as somatic embryogenesis. Multiplication of plants through plant tissue culture is known as micropropagation. Agitation is done in plant cell suspension tissue plant cell suspension cultures. 2 for all of them. Indole acetic acid is the first natural cytokinin. No, it's the first natural, it's oxen. It's false. The phenomenon of mature cell reverting to a meristematic cell forming undifferentiated callus tissue is termed redifferentiation. False, because it's dedifferentiation. Indirect somatic embryogenesis, the explant produces callus. which develop competence to produce embryos. Oh, should have come with indirects. It's true. Is it true? Yep, true. Good. Okay, week 3, pumping them out. Xylem elements are observed in the globular stage embryos. No, it's the heart stage later on. Preservation of genetic material in the form of seeds by providing suitable conditions in the gene bank is called ex situ preservation. It's not ex situ, it's in situ. It's false. Oh shit, I got that one wrong. Cell tissue cloning can be facilitated with the help of protoplasts, definitely. DNSO is a non-permeating cryoprotectant. False. Good. Phytoalexins are generally undetectable in a plant before infection. They are synthesized very rapidly after microattack. Bloody oath they are. What's the role of mannitol? That's a osmoticum. The probability of meristematic cells before free from being virus and higher is because of non-existing vascular connections in the plasmodesmata. Viruses cannot travel quickly enough through the plasmodesmata to keep up with the activating dividing cells. Plasmodesmata, that's the thing that links cells up. So there's a little cell there and cells, little joining, little bridge. That's that. What that's called. I'm not going to read all that. Oh, you can if you want. Later on, press the pause button. Which of the following is not a function of secondary metabolites? Well, that one. They are critical. That is not a function of secondary metabolites because they're not critical for the growth. That's right. Some plants release secondary compounds which have detrimental effects on the neighbouring plants. This phenomenon is called allelopathy. Oh, that's it. Week 4. Selection is an active process. Sure is, it's not a passive process. When a plant survives infection by a pathogen at one site, it often develops increased resistance to subsequent attacks at other sites throughout the plant. This phenomenon is called its induced wide systemic resistance. So it's false. Edioblasts can serve, yes, as secondary metabolites, storage places. There's so many places for storing the secondary metabolites. Adding pectinase in the medium 1 to 2 days before cell sieving may definitely disrupt large aggregates in the cell suspension. True. Microbial cystic molecular patterns are recognized by specific receptors, which then induces massive phytoalexin production in a defense strategy. It's referred to as acquired immunity. No, that's systemic immunity. Oh, that's amazing. Did you know that the microbes, did you know the plants can actually detect if they've got a not the felon, oh shit, the flagenta, the little tails at the back of the come out, the celiac type things that make them move in the microbes. The plants can actually distinguish if it's mobile or not by one of those. That's so cool. That come up in that section. Cell differentiator is a component of metabolic regulation in high organisms, definitely. Epigenetic instability causes genetic instability, leading to product accumulation, yes. Which of the following is not an approach to enhance secondary metabolite production? Well, they all do. See all the stuff you can do to enhance them, but microbial propagation. Salicylic acid, one of the 9 plant hormones, accumulates dramatically in the zone of infection after the initial attack. This sounds pretty right. It's systemic. Systemically acquired. And the vacuoles is very acidic. Vacuole is able to trap the ions due to its acidic environment. Okay, week 4. Week 5. Ooh. Something is a process by which functional groups of organic compounds are modified by living, so biotransformation. The process of cell immobilization by polymerization in a mixture of monomers in the presence of cell suspension is called entrapment. Something can be supplied, carbon dioxide can be supplied as a carbon source for photoautotropic cultures. Sorry, I shouldn't be saying, I'm saying it the wrong way. This is just like me training. Casein hydrolysate and peptone are used as a source of, in plant tissue culture, of nitrogen. The interactive effect in process parameters of the desired output in fermentation can be affected and optimized by using a response surface methodology. True or false? Immobilization can be advantageous in continuous process as it enables the reuse of cells till they remain viable. Bloody oath. That's the best bit about tissue culture. You can just keep pumping them out. It's so cool. Just got to have the right conditions for it. Say various smidgen, God, you end up with shit stuff like I can't do it in the winter, in the summer. It is advantageous to immobilize plant cells when the desired product is in non-growth. It's definitely advantageous. True. Sodium, yeah, I'll do my finger. Sodium chloride can be an example of a biotic elicitor. No, it's not bio, it's not alive. Failure in secondary metabolite yield enhancement upon addition of its metabolic precursor in the medium can lead to its diversion into active. Oh, it's behind my little finger. Alternative pathways. Failure in secondary metabolite yield can be had to diversion of, yes, true. Isopropanol can be used for cell. Yes, that definitely can. That breaks them up a bit. We'll put the little finger across again. Something is DNA sequence that increases the activity in the promoter and hence the likelihood of transcription increases. Enhancer. What's the likelihood of the reporter gene? Luciferase. Plasmid with a copy of 50 can be classified as low, medium, or high. Moderate. Which of the following generally enhances virulent gene expression in Agrobacterium species during plant cell infection? It's acetylsyringol. Something is the technique of direct gene transfer. DNA is delivered into the cell cytoplasm. That's mediated DNA delivery. Agrobacterium rhizogenes transfer something region into the host plant, which forms part of the RI plasmid. That's the tDNA. Sorry, I shouldn't even. Something vectors are used for making millions of copies. Well, we on a clone. Selectable markers are used in genetic transformation to prevent the growth of untransformed cells. Selectable markers. This must be true false because that's you want to use selectable markers. Yeah, true false. It's true. Hairy roots cannot grow on hormone free. True. Electroporation is an indirect gene transfer method. Indirect. No, it's a direct transfer method. So it's false. Top 10. Week 6. Woohoo, nearly finished. Oh, week 7. Holy moly. Which of the following factors make plants shears sensitive? If you throw them in a washing machine, the cell walls are going to be ruptured and there's large vacuoles. Identify the following impeller. Looks like a spiral, doesn't it? elliptical screw.
Elliptical screw.
Which of the following reactor designs have high aspect ratio? Like bubble. Bloop, 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 bloop. Which of the following is not an example of a gas phase reactor? Rotating drum and bubble. Yeah. Presence of suspended solids such as plant cells may reduce the KLA, which is the yield, by all of them. Formation of wall growth during cultivation of plant cell bioreactors may cause necrosis of cells leading to cell viability. Definitely. Orbital motion causes circular currents in airlift reactors. Orbital motion, no. Airlift reactors should have their own flow. Reactors in which liquid is dispersed, phase and gas is continuous is called a liquid phase. No, it's whichever's in the continuous phase. So that's a continuous, that's a gas. So no, that question should be a gas, that's wrong. The design of the impeller may not play a role in culture viability. Pig's ass, they make a huge difference. Photoautotropic cultures may require the presence of light jackets, definitely. Otherwise, how will they be able to get their light and make their chlorophyll action? And hexadane is generally used as a to improve the product from plant cell in a culture medium. Is a cell permeation enhancer. Spin filters in a bioreactor aid in keeping the cells retained. Mathematical based model approach is advantageous because shitloads of things, all of them. One of the statistical designs in experiments, which is used to screen significant parameters affecting design response, is given in variables with the processes whose processes are Plowden, Placard, Bullman. Which of the following is not a base or medium? Potato dextrose. Extracts of the same plant species collected from different agroclimates will demonstrate same profile of metabolites. Will they? No. False. Exposure time of a lithium can affect the product yield, the biomass. Exposure time of the lithium, can it affect? Definitely can. Steps to be formulated and active models are jumbled. Oh, this is the last one. Oh, this is that you got to go and put this that's into order. It's the mathematical model from doing the mathematics to formulate things. It just goes in the model where it's formulation of an equation, and then the second one is that's number two. Number two is determination of the model parameters, and then number three is evaluation of optimization and model parameters, and number four is parameter sensitivity tests and selection of significant parameters, and five is simulation of the experimental data. Sweet as, it's all done. And now we can go through some slides. These are rad. This is one of the very informative class. There's a hell of a lot of tissue culture stuff, but it does good on secondary metabolites too. It tells you, see your primary growth and then some secondary growth. So your primary growth is when it grows in length, and the secondary growth is when it grows thicker. So that's why you want to flip your plants, if anything, when you go, and at least they're a little bit thicker, so they're in secondary growth stage. Growth in meristems, that's different areas it grows in. Yep, your different meristems it's got there that we were talking about before. Now there's top, intermediate, and then apical. There's your root rams. and your shams, your root apical mass stems down the bottom. Cambium, cork layer inside the stem. I'll go through these pretty quickly because I reckon there's quite a lot of them. Culture in explant preparation, that's how to sterilise it. That's all the steps you should be doing. Callus induction, so as soon as you get your callus grown, that's a callus, a natural callus in a plant, and then that's one that's in the lab. And they're pretty easily to do it. You just add heaps of oxen and a little bit of cytokines, and you got yourself one, or any type of living cells. And you can make it out of dead cells, out of I'll go through that. Suspension culture, tissue culture. There's different techniques. This is pretty rad. Microculture chamber. So you can do, I did this on a slide. That's a slide for a microscope. You put two top glasses on the side, so it creates a little well in the middle, and then you can put it over the top. And then so you can put your plant underneath there and put a cover slip over the top, and it's all sweet. And then you can watch it grow. And that's how I watched it grow under there. And then if it grows, if you stay there long enough, because there's a lot of heat and shit, so you got to really watch it. But that's something fun to do. Plant cell viability. That's when you stain them. See, if you get some, I've got these stains, and you can see what cells are living and what are dead, and it only adheres to nucleuses of DNA in certain things or in proteins, and it's really cool. You can see what the viability in different types of cells is doing. That's for bacteria as well. Yeah, here you go. Look, natural requirements for plant cell. So when you're doing cloning, this is what cloning is. You're trying to start off something from nothing. So you need your inorganic nutrients, your carbon source, vitamins, growth regulators, and organics. The difference between your hormones and growth regulators, so growth hormones are produced naturally and growth regulators are added externally. There are your actual requirements. This is the actual full-on hardcore breakdown of them. So it's got everything that you need. Macros, your micros, your primaries and your secondary nutrients up the top there. And it goes in much more detail about them. Vitamin Bs. Vitamin Bs are so handy. Both plant growth regulators. Yep, that was what I was saying before. See how endogenous, that's why if they're done inside, and exogenous outside. All the oxidants and cytokinins needed. Differential organ regeneration. You can slow these up if you want. It's just shitloads of them. I want to get into the interesting ones. They're all interesting, but this is all on different types of tissue culture steps, things you can do, different ways you can do it. Somoclonal variation. A somoclone, that means it's just a tissue culture clone. So somoclonal variation means when it's got a tissue culture variation. Here we go, getting funner. Tissue budding. Methods of micropropagation. Look at those little micro balls, they're so cool. You can make them. These are the 4 shapes we're talking about the xylem. Remember it said in the globular shape, do you see the xylem? No, in the heart, or the torpedo, or the condylinas, when it's finally finished. They're 4 stages. Somatic embryogenesis. Let's go back and see if I was right. Xylem elements are observed in torpedo stage. Okay. This cannot travel. Yep. That's why it's one of those questions before. Correct amounts you're going to give it. Osmotic pressure. Protoplast culture is a cell missing the wall, like in oh, there you go. There's a protoplast. It's missing the cell wall. Animals have them. Easy to fuse and to mix with. Fusion procedures. Synthetic seeds, that is rad, those things. They're so cool. I got some of them. cryopreservation is very important. Excuse me. Methods. Yeah, you want to acclimate if you want it. When you do it, you just do it slowly. You can store stuff for ages. Acclimate it. Here we go. Secondary metabolism. This is cool. All the different stuff about this is so bloody in depth. Plant secondary metabolites can be divided into 3 different groups: terpenes, phenolics, and nitrogen-containing compounds. And we mostly deal with the first 2. And these 2 can be broken down into so much hardcore. It goes right down into everything. So your terpenes, and then you see in that and how many classes are involved in that. And in that, it's like shitloads of them. Terpenes named from there. Carbon chains. Out there. Anyway, who cares? Phenols, phenolic. They got that's where the carotenoids and flavonoids are involved. Anthocyanins, flavons and flavonins, isoflavones, rotenin. Rotenin's a really good natural insecticide. Alkaloids. Here we go. This all. Look at 15,000 alkaloids can be involved in the plant. And there was 10,000 of the I'm trying to think actually. Defense against insecticide and herbivores. This is how to undo your induced plant defense. So you can, I've done it with a few of mine, with my genes, and just introduce it into the lines. There's quite a few ways you can do it. And if you go through this slowly, you'll be rewarded like I have been. Strategies to invade them. Yes, infections, anti-plant, phytolexins, how they don't come out. Here you go. The microbe-associated general molecular patterns. The flagellin. That's what I was trying to think of before, the flagellin. See, they've got a receptor in the plant. It's the receptor FLS2. If that gets enabled in the plant, that means that the flagellin bacteria have been there. It's so cool. It's just, isn't it amazing?
Flagellins.
Yeah, so cool. That's how to do systemic acquired resistance. So you infect it, you just do a local resistance and it goes and throws through the stem. And the flower, let's get on with this, to induce it first. And it goes in through the stem. Infects it pretty decently. And it makes it, if it doesn't infect into that same, like if you've got it protecting you from not ladybugs, say from, I don't know, leaf miners, it'll build up a systemic resistance for that. But so if something else comes in next time, it'll have its proteins ready, so it can just go and activate it and do it quickly. So it's sort of primed, they call it. Accumulation.
Vaccine.
It's special compound. And that's what I got, some prime genes. Then you test them, you put them under those conditions and you test it, and then you see if it works. And I've done that too. Storage, yeah, these are all the storage plots for the secondary metabolites. There's a shitload of them. Vacuoles, blasts. Yeah, this is all the particular interest, commercial production of phytocompounds. So many of them. But they do so many good things. You can just go to those ones there and go and get all these anti-cancer drugs out of these species and varieties of plants. You get your disadvantage related to natural plant compounds. And they do this all through tissue culture. It's a real fast way to do it. And they get out micrograms at a time, but your micrograms on a large scale. Approaches screening selection. It's cool. Plating. Woohoo. There we go, at the end of week 4. Week 5 to week 8. Medium compound phytohormones, macro. Elicitors. Elicitors defined as molecules that stimulate defense or stress-related responses in plants. So elicitors, it could be a bug, it could be a wound that you've done, it could be from wind constantly on it. You know, an elicitor is an object or item or something that causes to stimulate the defense or stress.
Listen.
MERV gene, yes. Change your MERV gene. If you have Mediterranean disease, there you go, there's a fix for it. Get your doctors to go in and do that.
Mm-hmm.
Types of elicitors. Biotic and abiotic elicitors. Yep, there's a shitload of different ones. Yep. Stuff there. Emissions of biotransformation. So you want to use your elicitors to induce your plants with problems, so it'll back up their immune system and boost their genes. Mobilization. Entrapment. That's one of those questions. Viability testing. Let's see, here you go, do some viability testing. Stain, respire, cell growth. Genetic transformation in plant cells. So cool. Selection markers, it's really good to do that. And reporter genes, that's where that luciferase is. Plasmid-mediated gene transfers. Gene transfer methods. Plasmid, TI plasmid. Direct gene transfer and indirect calcium phosphate. Injection. There's no microinjection. They go directly inside the cell, inside a plant cell. You know how small that is, eh?
Yeah.
Just 1 to 2 microns. Here you go. This is pretty cool. I like this. This says the size of microbial cells are 2 to 10 micron and plant cells are 20 to 40 micron. Big difference. And then the culture time down here, 2 to 10 days for microbes, and for plant cells is 2 to 4 weeks. So that's pretty cool. And the growth rate doubling is when they double is 1 to 2 hours, and it's days in plants. So a couple of days. Really, really cool. Good comparative there. And it's some bioreactor differences. This is really cool. This is how to get different bubbles around. You want to keep it flowing so there's no dead spots in there, just different bioreactors designs to keep that happening. And that's how it says with this different reactors, propellers that you got, it makes them giant difference, because that's what that is. Some have, some haven't. Placed different heights, different angles, different widths, cut off here. It's fascinating. They make a giant difference. All this different design in slug bubble. See, blip, blip, blip. This looks like a slug bubble. Hairy roots. Oh, excuse me. Yep, they definitely need hormones. Advantage of hairy roots. Very fast growing. Gas phase reactors, hairy roots. Conclusion. Should come to the end.
Cells.
Show you case study. Oh, there's some assignments. Oh, do you want to have a look at it? A case study. What's the end? Objectives. This is a case study. So azadirachtin, which is neem seed, neem, the main constituent in neem, and this is a secondary metabolite extraction from plant tissue cell culture of neem. Neem seed azadirachtin. And that's when they went to the different parts of the country. They got all these different varieties. They grew them. They seen what different sounds they picked, and they picked out the best lines. It was from different areas, had different results. And they played with those lines and tried to enhance those lines and do different types of enhancement experiments that get different results. So then you can know by adding, your 2,4-D or some of, these different auxins, cytokinins, that it's going to give a different type of result. See over here, it's definitely changed. So you put a 4:1 ratio. Wow, it did really, really good of some indole butyric acid. And naphthalene and acetic acid did very good too. Anyway, it's just an example of, they went right through all this stuff and all different types of experiments. So if you're going to do like a PhD or something like that, this is what you would do. All listed as studies, which one works the best? Which one got the best response? They're listed as precursor addition. That's the precursor, is the thing that a chemical that you add that stimulates the chemical to be produced, like it's the precursor to that chemical being made. So if you go and add it, you'll get a really good process signaling down the line. So it can get upstart. So these are different types of them. Internalization agents they tried now. Large scale reactors. It's cool that they show you that everything, exactly what they put in. So if you want to replicate it or do your own experiments, you can just have this as a baseline. Inhibition studies. This class was very interesting. She was one of the most amazing teachers. She knows her stuff. She's not really reading much off the charts. She's just talking about experiments. And these are all her experiments too. So she can just talk and name everything quite quickly. That's interesting. That's it. All right. I think I'm going to stop the recording here. Hang on. How do I go and stop that? More. Pause recording. Stop recording. Thank you for watching. Hope everyone enjoys. Contact me if you want to talk about this, because I like talking about this sort of stuff. And if you're doing your weed, make sure you do it cleanly and smoothly and keep the health right up on it. Thanks, guys. See ya.